The greatest versatility in plasmid transfer is shown by the highly specialized Ti-Plasmids, Ti equals tumor-inducing, that allow certain bacteria to insert DNA into the nucleus of plant cells. The Ti-Plasmid is carried by soil bacteria of the Agrobacterium group, in particular Agrobacterium tumefaciens, and confers the ability to infect plants and produce tumors, inside which the bacteria grow and divide happily. This results in tumor-like swellings on the stems of infected plants, a condition known as crown gall disease. The related Ri-plasmid is carried by Agrobacterium rhizogenes, which infects roots and causes hairy root disease. Agrobacterium is attracted by chemicals, such as acetylcyranone, which are released by wounded plants. It then enters via the wound and transfers a portion of the Ti-plasmid into the plant cell by a mechanism similar to bacterial conjugation. A slight abrasion that is trivial to the health of the plant is of course sufficient for the entry of a microorganism. The result is a crown gall tumor that provides a home for the Agrobacterium at the expense of the plant. The Ti-plasmid consists of several regions, but only one segment, the tDNA, tumor DNA, is actually transferred into the plant cell, where it enters the nucleus. The tDNA is flanked by 25 BP inverted repeats. Any DNA included within these repeats will be transferred into the plant cell. Consequently, Ti-plasmids have been widely used in the genetic engineering of plants. The virulence genes on the plasmid are responsible for cell-to-cell contact and transfer of the tDNA but do not themselves enter the plant cells. Acetylcyranone, which attracts the bacteria to the wounded plant, also induces the virulence genes, thus facilitating the transfer of the tDNA region. Acetylcyranone binds to Vira protein in the Agrobacterium membrane. This activates VERG, which in turn switches on the other Vir genes, including VirD and VirE. VirD makes a single-stranded nick in the Ti-plasmid at the left border of the tDNA and the tDNA unwinds from the cut site. The single-stranded tDNA is bound by VirE protein and unwinding stops at the right border. The Ti-plasmid then replicates by a rolling circle mechanism as the single-stranded tDNA region enters the plant cell. Overall, this results in DNA transfer from the bacteria into the plant cells. The mechanism resembles bacterial conjugation and the virulence genes of the Ti-plasmid are equivalent to the trait genes of other plasmids. The tDNA then integrates at random into a chromosome in the plant cell nucleus. Once inserted, the genes in the tDNA are switched on. The enzymes they encode synthesize two plant hormones, auxin and cytokinin. Auxin makes plant cells grow bigger and cytokinin makes them divide. When this happens rapidly in the absence of normal cell differentiation, the result is a tumor. The tDNA also carries genes that subvert the plant cell into making apines. These are unusual nutrient molecules that are made at the expense of the plant cell but can only be used by bacteria that carry special genes for apine breakdown. The genes for apine degradation are found on the part of the Ti-plasmid that does not enter the plant cell. So the Agrobacterium can grow by using the apines but the plant cannot use them. Other bacteria that might infect the plant are also excluded as they do not have the apine breakdown genes either. Modified Ti-plasmids are widely used in the genetic engineering of plants. The genes for plant hormones and apine synthesis are removed and the genes to be transferred into the plant are inserted in their place. In practice, Agrobacterium carrying an engineered Ti-plasmid is used to transfer genes of interest into plants using plant tissue culture. In addition to inserting external genes into plants, the Ti-plasmid system may be used for analysis of plant gene function. Insertion of tDNA into the plant chromosome may disrupt a plant gene if insertion occurs into the coding sequence, or essential regulatory sequences. The model plant, Arabidopsis tholiana, has been used to generate a set of gene knockouts by random insertion of tDNA. The locations of nearly 90,000 such insertions has been determined, as of 2003. These include insertions into about 22,000 of the estimated 29,500 genes of Arabidopsis. These insertions may be used to investigate the functions of the inactivated genes by comparing the knockout mutants with the parental wild-type plant.